Okay, thank you. So I would also like to thank organizers for putting together this wonderful workshop, and also for all of you to stay here until the very end. So during this workshop, we already heard about like the swelling of sheets of hydrogels. We talk about pneumatic elastomers. So now we're going to talk about biological systems where sheets are growing, right? But here we're not going to have just individual sheets. We're also going to have sheets that are going to be interacting with soft substrates. And that's why we're going to get wrinkling, for example, in bacterial biofilms. And then I'm also going to tell you how in developing lungs, how we form like these new branches. And so before I go explain you how all of this happens, like you will see that there'll be like a lot of experimental slides here. And I'm a theorist, so this wouldn't be possible without the collaborative effort. So the wrinkling of bacterial biofilms was done together with groups of Bonnie Basler, Howard Stone, and Ed Wingreen. So Jing Yan was a postdoc who did most of the experiments. He was helped by a new graduate student, Chen Yifei. And my postdoc, Sheng Mao, also helped a little bit with the analysis. And the lung development was also done in collaboration with Celeste Nelson's group. And Katie Goodwin is a PhD student who did like all of the experiments. Initial modeling that I will show you in the end were done by a visiting master student, Tristan Guillaumard from ENS Lyon, who visited my group last year. And then Sheng Mao, my postdoc Sheng Mao, took over, and he is now continue working on these models. Okay. So we already, this wrinkling and branching actually happens in many places during the morphogenesis, during the development. So you all know that our brains are wrinkled, but maybe you don't know that all the wrinkles we get basically before we are born. So so these what you see here are CT scans of the fetal brain of the human. So like until the 20 weeks or so, like our surface of our brain is smooth. But then what happens after the 20 weeks? Because of the development of neurons, the neurons start migrating towards the cortex, and this effectively swells the surface relative to the interior, and this is going to create this is going to start creating folds and eventually these wrinkles. Maybe you're less familiar with the wrinkling inside the guts. So inside the guts, we have a lot of these like small pillars. These are called villi, and they are formed because we want to have like a large surface area, which kind of helps us absorb the processed food and send it to the bloodstream. And here, what you're looking is what you're looking is the cross sections of this gut. So this column shows you like cross sections along the gut, and this column shows a cross section across the gut, right? And so now we're looking at the chick embryo. And this is the age in days. What you see that initially the inner luminal surface is smooth, but eventually you start forming wrinkles. So first there are straight, smooth wrinkles, but eventually you also get zigzags. Because again, there's some differential growth and differential compression that's going to lead to these wrinkles. So today I want to focus on bacterial biofilms. As you can see, you can get like lots of interesting wrinkle structures in many different bacterial strains. And then, so this will be the first part of my talk, and in the second part of my talk, I'm going to focus on the lung development. So again, we're going to see how lungs gradually develop and form new branches. Yeah, so first it's really like differential growth, which kind of wrinkles the sheets. But once they are wrinkled, then these sheets actually start growing, and they will start protruding in the third dimension. So it's again another layer of growth. But it's like basically differential growth that leads to these shape transformations in all of these systems. Okay, so the basic mechanism was probably already addressed in this workshop, but just for concreteness, like what we're going to imagine is that we have like some stiff, thin, elastic film on some soft substrate, and in principle both of them can grow. And if they were not connected to each other, they would grow to their new lengths. And I will be working in a regime where the thin film will grow faster than the soft substrate. But if we force them to stay attached together, then what's going to happen if the mismatch of the growth is smaller than some critical threshold? Then the thin film is just going to compress and just going to be slaved by the substrate. However, if the mismatch of the growth is larger than some critical threshold, then this compressive stress in the thin film can be released by wrinkling, and this characteristic wavelength only depends on the thickness of the film and the ratio of the stiffness of the of the stiff film and the soft substrate, and this critical mismatch of and this critical threshold for the mismatch of the growth also only depends on the ratio between the stiffnesses of the substrate and the stiff film. So again, these are just asymptotic expressions that hold in the limit where the film is much, much stiffer than the substrate, but otherwise we get like more complicated expressions. Yes? Well, in the simple sketch, yes. When we're going to talk about lungs, we're actually going to have a full 3D thing. Okay. Okay, so now let's start with the wrinkling of bacterial biofilm. So what's going to happen? We're going to have like a soft agar, and on this agar we're going to put like a liquid drop which is going to contain bacteria. And now these bacteria are going to start growing, dividing, and they're going to start spreading on the agar. So this is what you're seeing. Now we're looking at this from the top view. So this initial liquid drop, and now it's gradually start growing, it's expanding. But now what you're going to see is that suddenly around now, like, you start forming these wrinkles, these radial wrinkles, and eventually, if you wait long enough, some of these wrinkles actually detach and they delaminate. So you see that here some of them are much darker, and those are actually the ones that delaminate. I'm going to show you some images of delaminations. And through this microscopy, we can actually get like the whole height profile of this structure. So, like often people will say, well, what's happening inside the core when you have like this mass? So remember, this is where the initial liquid drop was, right? So when I'm going to analyze the wrinkles, I'm going to ignore this internal messy region. I'm only going to focus on this radial region. Yes? So the typical height the typical height of this biofilm is of the order of like 100 microns. And single bacteria is like a few microns. So there are like several bacteria and so on, right? And this is a complicated process. Inside this biofilm, like we have bacteria that are actually growing and dividing. They produce extracellular matrix, which gives this film, which provides elasticity for this film. They also provide proteins which attaches strongly this extracellular matrix to the substrate agar. And it's a very complex process, right? And so it's not clear at all that this is driven by mechanical instabilities. That could be other mechanism that's driven here, right? I mentioned that some of them they also detach, and what happens like when this biofilm detaches, actually like all the dead bacteria are concentrated inside these delaminated regions, and while all the alive bacteria are inside this film, right? And it's still not clear. It's still like a bit of controversial whether like you first get the lamination and then that bacteria accumulate afterwards because they don't have maybe supply of nutrients, or whether first you have some bacteria that die and then this will lead to delamination. So it's still like controversial. Okay, so it's not clear at all whether these wrinkles that are occurring here, are they really coming from mechanical instabilities, or maybe they're just some result of some biochemical patterning processes, right? And I hope I'm going to convince you that at least the onset of wrinkles, I'm only going to focus on the onset, I'm not going to talk about the elimination, is really a result of the mechanical instability. Okay, so how did we try to convince ourselves into that? So first we're going to ignore the internal region. I'm going to now create a circle of some given radius r, and I'm just going to count how many wrinkles I have around the perimeter of this circle. And then I'm going to create like circles of different radii, and I'm just going to measure the number of wrinkles as a function of the circle radius. And what I see is that the number of wrinkles increases linearly, which suggests that the wavelength is constant, because like the perimeter increases linearly if the wavelength is constant. And you see here that you have like some bifurcations because we have to somehow bifurcate wrinkles so in order to accommodate more and more and more wrinkles. Okay, so the wavelength is roughly constant. That's great. We can also change the geometry. So instead of a liquid droplet, we can inoculate this biofilm with a razor blade. So now we're going to have like a straight biofilm that's going to grow straight. And again, we're going to get some wrinkles in this regime. So you can measure the wavelength of wrinkles for the straight biofilm or for the circle biofilm, and you see that they are the same. So really, the geometry of the biofilm does not matter. It does not determine the wavelength of this wrinkle. Okay, so we have done a lot more experiments. We have changed the concentration of the agar, which changes the stiffness of the agar. So we measured the stiffness in the rheometer. Perhaps this will also affect some other biochemical processes. So we have also measured the thickness of the bacterial biofilm. We have actually measured the shear modulus of the biofilm as well for all of these different cases. And then basically we can use the prediction from the elastic instability for what the wavelength should be, and we can compare this with the measured wavelength, right? So what you're seeing here is the measured wavelength from these images, where we're normalizing this with the thickness of the biofilm, and we're comparing this with the predicted wavelength that we get from the elastic theory. And what you see is that really, like the measured experiments really fall almost on top of this predicted line. You see that there are some deviations in this regime, but this is actually not surprising, because in this regime, the film is actually softer than the substrate, where this asymptotic formula really does not apply anymore. So, you know, hopefully this kind of provides some clues that, you know, this instability in this growing bacterial biofilm, at least the onset of instability, is really coming just from mechanics. Yes, Ben? So I think for the biggest mismatch is probably on the order of like maybe 10, but not more than that. Yeah. And here it's actually softer. Yeah. Okay, are there any other questions related to biofilm before I move on to lungs? All right, so now let's switch gears and let's look at the lungs, right? So again, we're going to look at the lung development in mice. And these are different stages of lung development, where this is the day of mice embryo, like age of mice embryo in days. And you see that the lungs initially start like this wishbone structure, but then gradually you start forming more and more branches, and in the end, like we get like these very complicated tree-like structures. And you know, like these new branches are formed through a series of these branching events. Either we split the branch of the tip of the branch and we create like two new branches, or we create like side domain branches. And this process is highly reproducible both in space and time. So these new branches will always form at the same location, and they will always form in the same sequence, right? So if we look at the lungs for mice at age 12, we're always going to see something like this, right? But we can also, so if we want to actually image of what's happening, we can actually surgically remove these lungs from the mice, and then we can just culture them on the plate for the next day or two before they completely die. But you know what we're going to see when we culture them, we still see some new branching events. And now we can actually try to see what is happening in here. Okay, so in this lung development, like there are like a few important players. So what's actually growing and branching? So inside the tube is like a luminar fluid, and then on the most inner layer of the tube is this like a single layer of cells. This is like epithelial sheet, and this epithelial sheet is surrounded by a soft tissue which is called mesenchyme. And this mesenchyme is going to provide all the chemical cues and chemical signals that are going to dictate the growth. And furthermore, some of these mesenchyme cells, they can differentiate and become these smooth muscles, which are these like red striped cells that you see here everywhere, right? And what is important here is that both mesenchyme and epithelium, they're much soft. They're a bit softer than smooth muscles. So smooth muscles are quite stiff, and they will basically guide the branching pattern, as I will try to show you. Okay, so here the red are smooth muscles, and in the green is epithelium. So for epithelium is ecadherin, and for muscles, I don't know, it's alpha sim something. It's basically some marker, fluorescent marker for these cells. Okay, I don't really know. Yeah, I don't really know. So like I asked Celeste, and she said that these are some typical values that people report in the literature, right? But they know that it's stiffer. But I know. Yeah. Yeah, well, like mesenchyme definitely is like viscoelastic, and I'm gonna mention this toward the end, right? So some of these tissues are viscoelastic, actually. Yeah. Okay, so let's move on. So like how now we would like to see like how all these different tissues cooperate together in order to form these new branches. Okay. So a few years ago, Celeste Nelson's group has already analyzed what's happening during these bifurcation events when we get like splitting of these two new branches. And what they see is that at the onset of the splitting of the branch, basically what happens at the tip of the branch, you get differentiation of smooth muscles, which eventually completely wraps around the tube, and then basically splits the tube into two new daughter branches. And what they see is that under normal conditions, they will get like two daughter branches, but if they chemically block the differentiation of smooth muscles, basically what they're going to get is they're going to get something which looks like wrinkled surface, because the epithelium sheet is growing relative to the soft background. Okay, so they already have analyzed this, but now we're going to focus on these side branches. And again, so here is again like lungs that were surgically removed from mice at day 11 and a half. And now this movie is going to play over the course of the next 24 hours. And what you're going to see there, we're going to start forming a new side branch right here. So now you see this is where the new side branch is forming. And again, we would like to understand, okay, so, what's happening that gives rise to these new side domain branches. Again, okay. Yeah. Yes. Yeah. Yeah. So definitely there is like a pressure difference. So there's definitely pressure inside the lumen, which you can think just basically swells the whole structures a little bit. Yeah. Yeah. Mm-hmm. No, it may not be, because you have like these smooth muscles all the way around that are controlling everything. Okay, so we would like to understand what physical mechanisms drive the formation of these new branches, right? And just like I showed you for the splitting of the two branches, here again they see that the smooth muscle which differentiate from the surrounding mesenchyme are important. So at the onset of the branching, initially the smooth muscles were covered on the left, but there was basically nothing on the right except that above the branch, which basically tells you like why it's branching towards the right and not towards the left. So this breaks the symmetry. But eventually, these stiff, smooth muscles, they're going to differentiate all the way around, and they're going to only leave a narrow opening for the new branch, where the new branch can escape and form. And again, under normal condition, we're going to get some new domain branch. So this branch was already there before, when the lungs were surgically removed from the mice, from the embryo. But again, if they chemically block differentiation of smooth muscles, they see that maybe they get like additional branches. They can also get, the onset of branches on the wrong side, and so on, right? So indeed, these kind of suggest that smooth muscles are important. Another effect that's happening here, beside the smooth muscles, is like all of these tubes, they're also growing, right? So now we can also quantify how fast they're growing and where they're growing. So again, they can chemically treat these tissues, and basically they can figure out, like at each given instant, like at a certain instant of time, they can mark all the cells that were about to divide, and this is sort of like a measure of some sort of measure of the growth, right? And what you see, like these like bright spots they mark all the cells that were dividing at that instant of time. That was like at day 12 of the embryo development, right? And here in the red, you see the epithelium layer, and in the green, you see the surrounding mesenchyme. And here, for now, we're only going to focusing on the growth of the epithelium, and we'll look at the cross sections where the new branch is forming, and another cross section just above and just below the new branch, right? So we're going to look at these cross sections, and we're going to try to quantify the growth of epithelium along these cross sections, right? So I'm just going to divide these cross sections into different sector angles. And then I'm just trying to quantify how many cells are dividing inside each sector. And then we can just plot the histogram, probability histogram, that the cells, probability histogram that the cells, probability distribution that the cells are dividing at different angles. And what you see here, that from this histogram, that we're in the region where the branch is, the cells are growing, dividing more on the right side than on the left. So they're growing faster on the right side where the new branch is forming. Initially, at the onset of the branching, at day 11.5, the same thing is observed just above and just below the tip. So again, you see more growth on the right side than on the left. But eventually, as these new smooth muscles are going to completely wrap around the tube, this is also going to block the growth, and then at the later stages, the growth is actually more uniform around these cross sections. So this just shows you relative amount of growth. But if you look at the absolute number of cells that are growing, actually here the number of cells that are dividing remains roughly constant, but here it actually decays with time because the new smooth muscles are forming, and you know, they can also block some of the chemical signals, right? So you know, this is now a complicated mechanism, like complicated system, where you know we have a lot of things going on. Maybe you know, like we said, like some smooth muscles are forming by differentiating from the surrounding cells. The tissues are growing, and they're growing unevenly, right? So you know, we would like to know what's going on. So in experiments, it's hard to control all of these things because you know, if we knock out something with drugs, you know, we can affect many things. So we wanted to create like a simple model where we could try to probe some of these issues. And I'm just going to show you some preliminary results that we have. Okay, so how do we model these growing tissues? Well, these things were already discussed when we discussed like swelling of sheets, right? So the idea is that you have some referenced undeformed state, and then you know we map this reference state into some final deformed state where the final deformation is a result of both the growth and elastic deformation. We basically have a map phi which maps from the internal coordinate system to the laboratory frame. And again, we can decompose this deformation into initial state, which is the growth, plus additional elastic deformation. And you know, this can be realized by decomposing the deformation gradient tensor, which are just derivatives of this map, and by decomposing this deformation gradient tensor into one part that comes from the growth and another part that comes from the additional elastic deformation on top. And it's only this elastic deformation gradient that's going to lead to internal stresses, right? The growth itself will not lead to stresses. So here, you know, what we're going to do is that basically, once we're going to prescribe the growth profile, we're just going to minimize the total elastic energy. Here, for simplicity, I'm just going to use Neo-Hookean model for all three different types of tissues, but we can also put in other types of models. So the results are quite robust and not really sensitive to the choice of this elastic energy. And we're going to have three tissues. Each tissue will have its own elastic constant. Like we said, stiff muscles, smooth muscles are stiffer than the surrounding mesenchyme, right? So here, for example, we have some example geometry when we have like a tube. So we have like a soft so this color shows the youngest models. We have like a soft mesenchyme. Here we have stiff, smooth muscles that maybe were there when we surgically removed the lungs from the mice. And the most inner layer will be epithelium that will be growing, right? And then, okay, so how do we describe this growth tensor, right? So like, you know, if we take like a small element of this cylindrical tube, then in principle, you know, like each of these elements will grow in three different directions. I'm going to, for simplicity, ignore any off-diagonal terms, which could in principle also happen during the growth. So in principle, we can have growth in the radial direction. So, so growth in the radial direction, which for the epithelium would be thickness, the growth in the longitudinal direction, in the axial direction along that axis, and growth in azimuthal direction. So in principle, like, you know, I'm going to prescribe these growth values for each of these tissues everywhere in space, and then I'm just going to minimize total elastic energy. And then I'm going to gradually increase the growth, and I'm going to see how the shape evolves. And for simplicity, I'm going to assume that the mesenchyme, the surrounding mesenchyme, is not growing. I'm only going to assume that the epithelium is growing. So epithelium is like a single cell layer, so I'm going to ignore the growth in the radial direction because, you know, it's a single cell layer. And you know, in principle, I can have both growth in the axial direction and in the azimuthal direction. Now, the movies that I'm going to show you, I'm only going to look at the growth in the axial direction, but everything looks exactly the same if I include also growth in the azimuthal direction. Okay, so now, first, I'm going to look at the case where there's going to be no differentiation of new smooth muscles. So all the smooth muscles were there from the beginning of the experiment. And now what we're going to look is we're going to have, so here we're going to look at the movie where on this color on this side you're going to look at the map of the Young's modulus so that you can see where the different tissues are. And here we're going to look at the stress map. And then, and also like there is no real dynamic, so this will be a movie, but there's no real dynamics in our system because we assume that the growth is very, very slow, so that we're always in quasi-equilibrium. So I'm just going to increase the growth. And like I said, I'm only going to assume axial growth, but it's going to be non-symmetric. So it's going to grow more on one side of the tube than on the other. And according to experiments, we see that it grows roughly 3 times faster on the right than on the left. So now our sheets start growing, and it starts a bit curving because it's growing more on the one side and then on the other side. But eventually, when the stresses become large enough, you get instability and you get wrinkles. And this was really like a full 3D simulation, right? So they're all the same movie. On the left, I'm showing the Young's model so they can see where these different tissues are. On the right, I'm just showing stresses, right? So you see that the stress is building up. There's larger stress on the right because the growth is faster, and that's why we get first wrinkling here, and eventually we also get some wrinkles there. So these sort of like mimic this like experiment where they were blocking smooth muscle differentiation, where they got like additional branches. The onset of additional branches, which is sort of like these wrinkling instabilities. Yes? Okay, so the compression comes, so basically the compression comes because so imagine they have a thin film that grows faster than the substrate, right? So if they were not attached together, the film would just grow. But because they're attached together, then the film has to, adhere to the substrate. So basically the substrate is compressing the film. Yeah. You get these zigzags, yes. Yeah, yeah, yeah. You can get like more complicated buckles, yeah. So, but here also like like the characteristic wavelength is actually quite comparable to the diameter of the tube, right? Yeah. Yeah. We have done that. Like these cross sections look actually very, very similar. There's not big difference because also the growth. Remember that the growth profile is larger on the right than on the left. So like the first onset of instability is only happen on this side, and then initially all these wrinkles will die out as you go around, right? So yeah, like you can get like more richer pattern, but it doesn't look much more different, right? But actually, like if you want to get the proper branch, that's not how you should do it because you're going to get too many branches, right? So these smooth muscles are really important, as I already showed you in experiment. So what I'm going to do now in my simulations, so now as I increase the growth, I'm also going to differentiate some of these tissues from the mesenchyme to become stiff, smooth muscles, right? So over time, we're going to get more and more smooth muscles. So I'm just going to assume that this is going to linearly change the elastic constant of my tissue in this neighborhood. So gradually, as I'm going to run the movie, you're going to see transition from soft mesenchyme to stiff, smooth muscles. And then we're going to leave one opening where the new branch can escape, right? And of course, the speed of this differentiation is very, very important, right? Because if this differentiation is very, very slow, then we're still going to get wrinkles. But if differentiation is fast enough, then you can suppress the wrinkles and get new branches. That's right. That's right. So for now, I'm going to ignore contractility, but contractility actually may be important, right? Maybe I'm going to return to that in the movie in the end, right? So now what you're going to see is another movie where now if you focus on these Young's modulus, you're going to see that this soft mesenchyme is going to turn into red. It's going to become stiff, smooth muscles, right? So now it's getting stiffer and stiffer and stiffer. And now all the wrinkles are suppressed, and the only way the new branch can escape is now through this narrow opening, right? So at least this is like another hint that these smooth muscles are really, really important, right? And sort of like this is where we are right now, and there are like a lot more open questions to be addressed, right? So first, for now I only assume that only the epithelium is growing, but actually the mesenchyme is also growing. In the images that I showed you when I had like these white spots, there are also white spots in the mesenchyme. So we actually have some data there, but we haven't analyzed it yet. The other thing that I already mentioned, the mesenchyme is actually not elastic, it's actually viscoelastic. And they know that on the scale of 24 hours, which these things are happening, the viscous effects do start to show up, right? So we can try to update our model to try to put in this viscous effect into a model. And now what you mentioned is, they also think that the smooth muscles are not just passively providing like this stiff environment, they're actually contracting this. So maybe if I go back to the movie that I showed you earlier. Okay, so look at this branch, where it's going to form. Initially, okay, what just happened? So initially it's going to be very, very wide. So like look at the cross sections. It's very, very wide, but in the end it's going to shrink. So look at this. Okay, it's not, maybe I cut the movie too fast. So in the end, this is quite wide, but now this is going to start shrinking. And they think that this shrinking is coming from the contractility of the smooth muscles, right? So this is indeed another thing that we can try to put in the model, right? So we can say that once the smooth muscles form, they basically contract, right? Okay, and eventually, like it would be nice to, and the potentially there is also some mechanochemical coupling, like stresses can affect chemistry can affect the growth, and so on. So eventually it'd be nice to actually figure out what are the relevant morphogens that are driving this whole thing to come out with a complete story, right? So here I will end, and like I said, this was a very collaborative effort, and I would like to thank you for your attention and take your questions.